Good morning.
Yes, good morning. So today we are going to start with a new topic in instrumentation. Okay, and the name of the topic is polymerase chain reaction. How many of you have ever heard about this technique?
Yes,
Yes, sir.
PCR.
Yes, sir.
Yes,
Yeah,
sir.
yes. Yeah. So let me share my screen to you. Okay, can you see me? Can you see the screen?
Yes, sir.
Okay.
Yes, sir.
Yes, sir.
Uh, Okay, so that is the polymerase chain reaction. Polymerase, as the name indicates, it is an enzyme which can carry out the polymerization reaction, essentially of the DNA. Okay, and what we can do with this technique?
Amplification of DNA.
Yes, we can amplify a small number of DNA strands which are available to us. Okay, double-stranded DNA, of course, and then this DNA is amplified to a great extent, and we get a large number, or maybe millions of copies of the DNA are available, and then this DNA which is amplified is then used for different applications. Okay, so we start with a small number of DNA molecules and end up with a large number, and all this amplification is done in an in vitro, using an in vitro technique. Okay, so in the laboratory we can, without the use of cell systems, okay, without using any biological cell as such, we can convert a small number of DNA molecules into a large number. Okay, the name of the scientist who has discovered this technique is Gary Mullis. I think you probably have heard his name. He got the Nobel Prize for this important discovery. Okay, and this is the is in his own words. I have this sentence is stated here. Carrie Mullis was actually traveling one day. He had gone for a long drive in the middle of the night. Okay, and while he was driving his car, he was alone in his car, and he was trying to imagine something. Okay, and trying to calculate, and he had mentally he was in the process of trying to imagine how this particular reaction would go. Okay, and then suddenly he got very excited when he understood that a large number, millions of copies of DNA can be made using a very, very simple technique. And therefore he stated this line. Okay, it was difficult for me to sleep that night with deoxyribonuclear bombs exploding in my brain. Okay, if you can ever get a chance to read his autobiography, then that will be very good thing. It's written in a very interesting fashion. Okay, and this particular article comes in a scientific magazine which is called Scientific American. How many of you have seen an issue of Scientific American anytime in your library? Did you have this
No, sir.
in the college
No,
library? No. Okay, we have
no,
this
sir.
particular magazine in our library in the Institute of Science. Okay, so whenever you get an opportunity to come here after the lockdown, of course, then you will be able to go to the library and see this volume. Okay. But interestingly, 20 years ago, this idea was already proposed by an Indian-born scientist. Okay, Dr. Hargobind Khorana had already conceived this idea. So that is a thing of pride for all of us. Okay, I'll go to my next slide. How many of you can tell me what is this reaction, polymerase chain reaction? Anything that you have heard about it anytime?
Uh,
Can you see my screen clearly?
Yes.
फर्स्ट प्रोसेस डिनेचुरेशन का होता है, एनेलिंग, एक्सटेंशन, etc.
Denaturation of bond.
I didn't get you properly. Denaturation of what?
Denaturation, annealing, extension.
Yes, very good, very good. So I think you
Bonds
have
in
already.
strand.
Yes, yes. Okay, so this is the photograph of the scientist Carey Mullis, born in 1944. That is before the Indian independence. Okay, and he shared the Nobel Prize in 1993. Nobel Prize in Chemistry with Michael Smith. Okay, and then this invention was so important that it was described as something which virtually divides biology into two epochs. One is the before PCR and the other is the after PCR. Okay, so after PCR, a lot of things happen in biology. That was only because of this particular technique invented by Carey Mullis. Okay, so this is basically a very simple and rapid method of amplifying a small length of DNA, okay, which we call it as the target DNA. And this target DNA may be a few molecules to maybe 100 molecules, and that is amplified to thousands or lakhs of copies, okay. And ultimately we get about say 10 raised to 9 fold increase in the number of DNA molecules, okay. And that all happens in a test tube in a small vial, and that requires only a few hours. Okay, so it is a chemical technique, or rather more specifically, we can call it as a biochemical technique because enzymes are involved. Okay, the DNA polymerase enzyme is involved. So we can call it as a biochemical technique. Okay, so and it is a repeated synthesis, okay, repeated enzymatic synthesis of a segment of DNA. And the advantage here is that once we get a large amount of DNA amplified, then this can be used for other different studies. For example, we can characterize the DNA, we can sequence the DNA, and then we can probably use that DNA for cloning experiments. Okay, we can insert the DNA in different types of cells. It may be biological cells or yeast cells or whatever it could be. Okay, and this essentially makes use of a specific enzyme called DNA polymerase. And then we also require something called as the primers. Somebody said that annealing is a step involved in PCR. Okay, so these primers are what are attached to the target DNA at the extremes, 3' end of the DNA. And then these primers are, they serve as a starting point for the synthesis of new DNA molecules. Okay. Can you all see this diagram here? Okay, we have the target DNA here, the two strands of the DNA, the 5' to the 3' end of one strand, and the 3' to the 5' end of the another strand. Okay, now these two strands are separated. That is what we call as a denaturation step, which happens at a high temperature of more than 94 degrees. Okay, so when the two strands are separated, then the next step is the annealing of primers. So can you see here a small fragment of the primer is there? Okay, and this primer is annealed to or joined to the 3' end of both the strands, one here and one here. Okay, and then the DNA polymer, this is called as the annealing step, which requires a little lower temperature. Okay, 50 to 55 degrees. And then in the third step, starting from this particular end, 3' end of the primer, we have the incorporation of nucleotides, okay, in the growing chain from the 5' end of the primer to the 3' end. We get the synthesis of DNA along both the strands of the DNA. Is it okay? And therefore, both the strands now will have one old and one new strand. Okay, and one particular molecule of DNA is now converted, or it is amplified into two molecules of DNA. Is it okay? Okay, then about many thermal cycles are repeated. Okay, so we call this term as cycles because the same steps are repeated again and again. What are the same steps? There are three essential steps. One is the denaturation, the other is annealing of the primers, and the third one is the amplification. Okay, that is the synthesis of DNA. These three steps, the first steps requires 94 degrees temperature. Okay, the second step requires about 50 to 55-degree temperature, and the third step of DNA polymerization, that requires about 70, 72 degrees centigrade. Okay, so these three steps are repeated again and again, say for up to 30 times or 35 times, and at the end of the 35th cycle, probably we get a large amount of the DNA which is synthesized. Okay, step number one is written here, that is dissociation. It requires about 20 seconds. Okay, and in this particular step, the two strands of DNA get actually separated from each other because the temperature is high. That is called as the melting temperature. At the melting temperature, the two strands of the DNA will get separated from each other. And for this, we require, say, about 0.1 microgram of DNA, the target DNA, and that comes to about, say, 100 to 1 lakh copies of the target DNA. Okay, and sometimes even less number of copies are also enough for the DNA amplification to take place, right? This step occurs at 94 degrees. Okay, although 94 degree is not a very specific number, but it could be, say, few degrees or less or few degrees more also, depending upon the sequence of the DNA, right? Step number two again takes about 20 seconds, and that is the annealing of two primers at the two extremes of the target DNA, that is at the 3' end of the target DNA, right? And these particular primers will anneal, or they will join by complementary base pairing to the target DNA. And this will happen at a lower temperature of about 40 to 60 degree, but usually it is, say, 50 to 55 degrees centigrade. Okay. Third step takes a little longer time, that is 30 seconds. And in this particular step, the polymerization reaction happens, that is the DNA starting from the primer, new DNA synthesis will take place. Okay. And for this, the temperature which is required is about 72 degrees. The total amount of time which is required for each cycle is about 3 minutes and 45 seconds. Okay. The number of cycles required, of course, will depend upon the number of copies of the template DNA or the target DNA which is available. The reaction volumes are usually 50 microliters to 100 microliters. Sometimes it could be even 25 microliters. Okay. And what does the reaction mixture contain? That is very important for us from the standpoint of actually carrying out an experiment. Okay, so we have about 0.1 microgram of the target DNA or the template DNA. Then we require a set of two primers. Okay, and these two primers are going to anneal to the two ends of the DNA, and the concentration of primers required is about 0.25 micromole. Okay, micromole of DNA, and this DNA is of a known sequence, that is very important. Okay, the sequence of the primer is known to us. And how they are known to us, why they are known to us, because the extremes of the DNA should also be known to us. That is also known, and therefore we can select the appropriate primer which will anneal to the extremities of the DNA. Okay, then the DNA polymerase is required about 2.5 unit of the DNA polymerase. Now I have one assignment for you. Okay, the assignment is this, that you should try to define what is 1 unit of DNA polymerase enzyme. Okay, I think in one of the last lectures sometime I have asked you about the question, what is 1 unit of enzyme activity, and some of the students did answer me correctly, and they said that it is the amount of the enzyme which is required to convert 1 unit amount of the substrate, say 1 milligram of the substrate or 1 microgram of the substrate, to the product. Okay, in a given amount of time, say 1 second or 1 minute or whatever. Okay, so on a similar line, you have to tell me what is the definition of 1 unit of DNA polymerase enzyme. Did you get my question, everybody? हेलो,
sir.
हाँ कौन बोलते हैं? Who is that? May I know? Meenal, Meenal, are you there?
Yes, sir.
Okay. Could you understand my question? What is the question? Hmm? My question is, you have to find out what is 1 unit of DNA polymerase activity.
So please repeat the question.
Yes, the question is, what is the definition of 1 unit of DNA polymerase activity? Okay, how do you define 1 unit of DNA polymerase activity? Is it okay? After the lecture is over, you can go to the literature, you can find out from the net, or you can find out from any book if you have. Okay. Yeah, then we go ahead. In this reaction mixture, we also have to add the raw material. Okay, so the raw material here is the 4 types of deoxyribonucleotide triphosphates, that is DATP, DGTP. What is ATP?
Adenosine
Triphosphate.
Yes. Adenosine triphosphate. GTP is, what is GTP?
Guanine
Guanosine triphosphate. CTP is
So cytosine triphosphate.
cytosine triphosphate, and
Cytosine
TTP,
thymine triphosphate.
thymine triphosphate. Very good. Okay. So the concentration of each of these deoxyribonucleotide triphosphate is 200 micromole. Okay. Just remember that. Then in addition to this, we also require certain conditions. Some ions are required. Okay. So KCl at a concentration of 1.5 millimole. Then MgCl2 at the concentration of 1.5 millimole, and this is all taken in a buffer, okay, because the conditions must be maintained. The enzyme should remain in a proper active form, okay. So the buffer uses Spritz HCl of 10 millimolar strength, and the pH of the buffer is about 8.4, okay. And then we also add gelatin, okay, gelatin 100 microgram per ml. Why is gelatin added? Gelatin gives a certain amount of viscosity to the medium.
Silting down.
Yes,
Silting down.
I didn't understand you. What did you say, Shamim?
So let's settle down.
Settle down. What should settle down? I think you made a good attempt to answer. I think you are trying to take something from the electrophoresis experiment. I told you in electrophoresis we use sucrose in the sample buffer.
Sir, yes, sir.
Okay, and that sucrose is used so that the sample should go
Yes, sir.
within the wells. It should not come out of the well. That was the reason. Okay, but the reason why we use gelatin here is
Yes, sir.
it's a different reason. Okay. In fact, here we don't want anything to go down at the bottom. We want everything
Okay,
to
sir.
get mixed properly. The reaction should happen in the buffer, isn't it? So we don't want any component of the reaction mixture to go to the bottom of the container, to the bottom of that vial, right? So gelatin here is to mimic the conditions that are present in a real cell, okay? Because usually in the cell, okay, the DNA amplification or DNA replication is happening in a biological cell, okay, in the cytoplasm, in the nucleus, isn't it? Am I right? Okay. The conditions in
Yes.
the cell are such that there is a certain level of viscosity. Okay. The enzyme should feel that it is in a cell, in a cellular environment. Okay. So in order to mimic the cellular environment, we use some amount of gelatin. Is it right? Have you understood?
Yes.
Okay.
Yes, sir.
And then the question is, where is this PCR reaction carried out? Okay, there are two things we can do. The reaction is either carried out manually, okay, and this is carried out in water bath. So essentially we have vials which contain the reaction mixture, and then these vials are kept in water bath, okay, and the temperature of the water bath is adjusted to the desired temperatures. For example, we require 94 degrees for melting, we require 55 degrees for annealing of the primers, and we require 72 degrees for the polymerization reaction or extension reactions. Okay, so these temperatures are maintained in water baths, and then the reaction can be carried out within the water bath. That is one thing. And the other is we can use an automated machine which is called as the DNA, which is called as the PCR machine, or it is also called as the thermal cycler. Okay, thermal cycler machine. Okay, PCR machine or thermal cycler machine. Both the names are used. Is it okay? And in the
Yes, sir.
next lecture, we are going to study the details of this machine itself, how the machine works, how the machine is constructed, and things like that. Okay, how it is used, right? At the end of these cycles, okay, each cycle, the number of DNA copies increases exponentially, okay, and therefore within say about 2 hours time, there are more than 2 raised to 30 copies of the DNA, 2 raised to 30. And that is about 1 into 10 raised to 9 copies of DNA. Okay, so how much is 10 raised to 9? 10 raised to 9 is about 100 million. Is it right? 100 million copies. Right?
Yes.
And I'm getting some message here. Don't know why. If you are interested, then I'll just tell you one information, and that is we know that there are too many stars in the universe, isn't it? That the number of stars which is estimated by scientists, okay, in the total universe, that is about 10 raised to 21 stars in the universe. Okay, and then can you compare the number of DNA molecules which we get in one vial? In one vial, we are getting about 10 raised to 9 copies of DNA. Okay, so in order to get 10 raised to 21 copies of DNA, how many reactions do we have to carry out, PCR reactions? Or how many vials do we require so that we will get a total number of DNA copies that is equivalent to the number of stars in the universe? Can you do this calculation briefly?
Two-
Hmm? Do you have a notebook and a pen with you?
2.5.
Yes, then try to calculate this. Simple arithmetic, no?
So 2.5 about.
What, 2.5?
No. 4.5. About
What is
4,
it?
sir.
Wha-
4
wha-
hours.
No, I'm not talking about hours. I'm talking about how many vials will be required or how many reactions we have to carry out so that we will get a total number of DNA, which is about 10 raised to 21. Okay. In one reaction, we get 10 raised to 9 copies of DNA. So how many reactions we have to carry out to get 10 raised to 21 copies of DNA? I think I have 10 minutes for this lecture. So after 10 minutes, I'm going to end this lecture, and then the next lecture will be engaged by TK Cursor. Okay, I have already put the message in the on the WhatsApp. Okay, maybe we'll get about 5 to 10 minutes break in between, and then TK Cursor will continue. And another thing which I wanted to ask you is about the lecture we had about Dr. Kailash Chaudhary had given one lecture on how to crack the competitive exams. How many
Yes,
of
sir.
you attended
Yes.
that lecture?
Yes, sir.
Sir, we attended on YouTube
Okay. Yes.
because
Yes.
the slot was full.
Yes. Yes. Okay. No problem. So Inamul attended. Joshi Shraddha attended. Who else attended?
Yes, sir. I'm attending.
Sir, I'm also attended. I'm attended Zoom meeting.
Okay.
Sir, मी Zoom वर attend केलं sir.
Okay. But why did you not make a comment in your on the WhatsApp group? You should have written something. Amol Pawar, you also attended. Okay. You can raise your hands. Okay. If you have attended, please raise your hands.
Sir, I also attended.
Raise your hands. तुम्हारा येता का raise your hand करता याच्यामध्ये?
Sir,
ओके एक मिनिट
yes, sir.
हे कसं बघायचं बरं? I don't know how to see that. Okay, हाँ. शमीम जोशी
Yes, sir.
श्रद्धा, प्रल्हाद हुंबे, अमोल पवार, इनामुल हक, प्रशांत गिरे. अजून कोणी केलंय? शीतल जाधव, could you attend that lecture? Sheetal Jadhav,
No, sir.
you should have attended it, but then I think it is on the YouTube. Okay, so anytime you can see that lecture. Okay, you can attend it. Is it okay? It was a very, very
Okay,
useful lecture. Okay. And those who have already attended the lecture, you can just give a comment in the WhatsApp group. Okay, because we want to know your feedback, and based on your feedback, we will that will also inspire us to organize more such programs in the future. Okay.
Okay.
So how many of you have
Yes, sir.
done the calculation? See, I'll ask you a simple question. If I can have 10 raised to 9 copies of DNA in one reaction, then in 2 reactions, how many copies of DNA will I get? Any answer?
18.
10 raised to 18.
8 is 2.3 sir.
10 raised to 18. Is it right? 10 raised to 18?
Yes.
Just a minute. Let me go to the board. Okay, whiteboard. In one reaction, I'm getting 10 raised to 9 copies. Okay, so 10 raised to 9 means like this: 0, 0, 0, 0, 0, 0, 0, 0, and 0. Is it correct? 10 raised to 9. Okay, and if I have 2 vials, then it is 10 raised to 9 plus 10 raised to 9. Okay, if the meeting gets over, I'm just telling you that I'm not going to start the meeting once again. Okay, after 10 minutes, Tikekar sir will engage your lecture. This calculation we can do later. Okay, so how much is this total now? Is it like this? One, two, three, four, five, six, 7, 8, and 9. Am I correct? You can just correct me, okay, if I'm wrong. Okay, so this number actually is 2 into 10 raised to 9.
Nine.
Is it right? So if I have it like this,
Yes, sir.
if I repeat it 10 times, if I carry out 10 reactions, then it will be 10 into 10 raised to 9. Okay, which is equal to 10 raised to 10. Is it correct? Okay, so 10
Yes,
reactions,
sir.
10 reactions is 10 raised to 10, right? So if I want to make 10 raised to 11, how much is it?
Sir, 11 times.
20 reactions. Is it correct? Am I correct? 20 reactions is 10 raised to 11. 30 reactions is 10 raised to 12. Is it correct or am I still wrong? You have to correct me.
Sir, I think 11 for 10 days to 11.
For 10 raised to 11, what do I have to do?
No, no, sir. 30 reactions for 10 days to 30.
Is it correct? 30 reactions? Or is it wrong? Or it should be 300 reactions? Anyway, so this is the kind of calculation I have given you for your homework. Okay. Tomorrow and day after tomorrow, by Sunday, you try to solve this question. Okay. And everybody should answer me in the next lecture. Is it okay? Can you do it?
Yes, yes, sir.
And then I just want you to you all to appreciate this fact that as many stars there are in the universe, those many DNA molecules we can make. The humans are so intelligent that humans have devised a technique for amplifying the DNA to the extent of the number of stars that are there in the universe. Okay, so that is the only thing which I wanted to tell you. Is it right? Okay, 12:30 lecture will be engaged by Peshwe madam. Okay, everybody, just give attention to this announcement. The next lecture will be at 12:30, and it will be engaged by Peshwe madam.
Okay.
Okay. Are there any questions?
No, sir.
Hello? No questions? Okay, then
No,
we'll
sir.
stop the meeting. Okay, and wait for madam. 12:30. At 12:30, madam will engage the lecture. Okay.
Okay, sir.
Okay.
Yes.